You guys know what proteins are? Cool. Okay. All right. Okay, so that guy on the left, okay, is a picture of hemoglobin. Okay, hemoglobin is the protein or sort of tetramer that carries oxygen around the blood. And you'll notice, if you feel like it, that those guys, there's shapes here that you may or may not be able to see that are heme groups, and they have iron in them, and they bind to oxygen and carry it around your blood. And you'll learn in biochemistry, those of you who are going to take that, all about the mad, amazing workings of this thing and when it goes wrong. Okay, so this is an illustration of the 3D structure of hemoglobin. Okay, proteins' workings and the workings of enzymes, which are special proteins that are like machines, depend in part on their three-dimensional structure. Okay, but when a protein is made, it's not made as a 3D thing, it's made as just a string of amino acids. Okay, that string of amino acids spontaneously, usually spontaneously, folds up into that three-dimensional shape. Okay? And it would be good to be able to figure out, given the sequence of amino acids, what shape you're actually going to get. Okay? In case you want to design a protein that's a drug, okay? Or you just want to analyze something. So you know what the sequence is from a gene or whatever, and you want to know what shape that thing's going to take, so what it's going to do. And the question is, how do you do that? Okay? And in fact, what does the protein do? How does it fold up? Okay? Typically, it folds up spontaneously, and what that is supposed to be is an energy landscape. And what happens is, as this thing folds up, it folds and it fiddles, and its free energy changes. Okay? And things are lazy. Physical systems are lazy. They want to go to the lowest possible energy. So what this thing does is, as it's made, it folds up and fiddles around, and largely spontaneously, and winds up going to the lowest possible energy. Okay? So again, that's the energy landscape. You can picture it bouncing around in there as it folds up around. And it gets stuck momentarily in little local traps, but eventually, hopefully, finds its way down here. And down there is the lowest energy state, and it's the native state of the protein. It's what the protein's going to look like as it's folded properly. Okay? And if we're going to design proteins or analyze proteins, we want to figure out what that looks like, okay, without having to do an X-ray structure every time. So what you'd want to be able to do is to figure this out on a computer. Okay, so there's a big field of trying to figure out what proteins look like and trying to design proteins and all this just from their sequences, okay, by finding the sort of conformation in which they'd be in their minimal energy, so what their folded forms would look like. Okay, so again, this is an optimization problem. Find the minimum energy state of the protein, that's what it's going to look like when it's folded. Okay, and this is a ludicrously difficult problem. Okay.